Hello, this is Matt Dean with A+ College Ready, and today we're going to talk about non-Mendelian inheritance. Although Gregor Mendel discovered some of the most basic laws of inheritance, scientists have now discovered many exceptions to the so-called laws of Mendelian inheritance. The processes and conditions described in this presentation are all examples of non-Mendelian inheritance patterns. So let's start off by talking about linked genes. Linked genes are genes that occur on the same chromosome, and therefore usually they tend to be inherited together. They don't segregate. One of Mendel's laws was the laws of segregation. When two heterozygotes are mated in a normal dihybrid cross without linked genes, with independent assortment, we get an expected phenotypic ratio of 9:3:3:1. That's Mendelian inheritance. However, as shown in the figure on the right, in the case of dihybrid crosses involving gene linkage, the ratio of the expected offspring produced is supposed to be about 3:1. So in this case, we're looking at long wings and red eyes. So we have an organism that's homozygous for long wings and homozygous for red eyes, crossed with an organism that's homozygous for short wings and homozygous for white eyes. And we can see from the diagram here that the long wings and red eye gene are on the same chromosome. So the genes for length of wings and color of eyes are linked. When this organism goes through meiosis and makes gametes, all the gametes should be big L, little r. When this organism goes through meiosis and makes gametes, all the gametes should be little l, little r. And therefore, in the F1 generation, all the offspring turn out big L, little L, big R, little r, and they all have long wings, which are dominant, and red eyes, which are also dominant. If then we take two of these offspring and cross them together, so if we do a big L, big R, little r, crossed with the same thing, we would expect under Mendelian inheritance to get a 9:3:3:1 phenotypic ratio. But because of the linkage of the genes, what ends up happening is that, let us back up for a second. If we were thinking about Mendelian inheritance, this organism ought to be able to make 4 genetically different types of gametes. Big L, big R, big L, little r, little l, big R, and little l, little r. And this one ought to be able to make the same. So if we were doing a Punnett square for a cross between those, should be a 16-box square, 4 rows and 4 columns. But because of gene linkage, this can't happen in this situation. Because the big L and big R genes were originally on the same chromosome, they're still linked together. So this is a possible gamete. Also, the little L and the little R alleles were linked to the same chromosome. So this is a possible gamete. These two don't get made because of linkage. So instead of a 4x4 Punnett square, we're going to end up with a 2x2 Punnett square. And we end up with an expected ratio of 3 long wings, red eyes, and 2 short wings, white eyes. Now, the table on the right includes the actual observed results of the cross I just described. And notice that even though we didn't expect it, we ended up with some organisms with short wings and red eyes and long wings with white eyes. Mendelian inheritance doesn't explain that. Gene linkage doesn't explain that. What does explain it is that every now and then, those two genes that were linked together end up on different chromosomes because of crossing over. Sometimes the genes go through recombination. Crossing over happens in prophase 1 of meiosis, and we will occasionally end up with some organisms that don't fit either the Mendelian prediction or the prediction due to gene linkage. So if you see some weird gene outcomes in a Punnett square, it's probably due to both gene linkage and also the crossing over that can happen with those linked genes. Another example of a type of non-Mendelian inheritance is incomplete dominance. So this is a form of intermediate inheritance in which one allele isn't completely expressed over the other allele. The example that people always go back to are in certain kinds of plants, like snapdragons, for example. There are two alleles for flower color, a big R, which is for red, and a little r, which is for white. Organisms that are big R, little r, are always going to have red flowers. Organisms that are little r, end up with white flowers. But organisms that are heterozygous, have one big R allele and one little r allele, they end up with an intermediate phenotype, pink. So sometimes you'll hear incomplete dominance referred to as blending, because what ends up happening is that the phenotype you get when you have a heterozygous organism, it's usually intermediate between the two main alleles that code for it. So red plus white gives you pink. Now, something that people often confuse with incomplete dominance is codominance. Completely different phenomenon. This is a condition in which both alleles are expressed at the same time. This is not blending because both of the alleles are equally present in the phenotype. The most common example that you'll hear about is human blood type, the ABO blood type. For human blood type, there are three different alleles. There's an allele for A, there's an allele for B, and there's an allele for O. The alleles for A and B code for different cell surface proteins on the surface of your red blood cells. Individuals with two O alleles don't have either of those. The O allele doesn't code for either protein. Individuals with two A alleles or one A and one O allele end up with the type A proteins on their red blood cells. Individuals with two B alleles or one B and one O allele end up with the type B proteins. But individuals with a type A and a type B allele end up with both the A proteins and the B proteins. They don't end up with a protein that's intermediate between A and B. They end up with A and B proteins separately. Another commonly cited example of codominance is an inheritance pattern of coat coloration in some cows. So there's two unique alleles. Big R is the allele for red coat coloring. Big W is the allele for white coat coloring. So if a cow has two big R alleles, the pure red. Two white alleles, pure white. But if a cow ends up with one red allele and one white allele, they end up with this coloration pattern. This is called a roan. So we're ending up with a distinct, the distinct expression of both the red allele and the white allele, not something in between. We don't end up with pink in this case. We end up with patches of red and patches of white. This is codominance. Another example of non-Mendelian inheritance is the presence of multiple alleles for a particular trait. In Mendelian inheritance, there were two alleles for each trait. For example, there was a tall allele and a short allele, or a purple flower color allele and a white flower color allele. But what scientists have found in some situations, there can be more than two different alleles for a particular gene. More than two different alleles that lie at the same locus or location. Each organism only gets two of those alleles, but there can be more than two different possibilities. For example, the example we looked at just a second ago in the ABO blood system, there are three different possible alleles. There's the allele for A, there's the allele for B, and there's the allele for O. Each individual only gets two of those, but they can get any combination of two of those. Another non-Mendelian inheritance pattern we want to look at is pleiotropy. Pleiotropy is a condition in which one gene affects multiple seemingly unrelated characteristics. Sickle cell disease or sickle cell anemia is a good example. The gene actually affects the shape of one of the polypeptide chains that make up hemoglobin, the protein in our red blood cells that helps us to transport oxygen. The change in this polypeptide shape, though, ultimately causes the red blood cells to change shape. from round to sort of sickle shaped. This impedes the flow of blood and leads to symptoms all over the body, that symptoms ranging from anemia to weakness, physical weakness, to impaired mental function, to lowered disease resistance, and even ultimately to paralysis. Now, something really interesting about sickle cell disease is individuals who are heterozygous for the sickle cell mutation get resistance to the protozoan that causes malaria. Now, this doesn't mean they're immune to malaria. They might still get it, but if they do, they're more likely to get over it and survive it. This is called a heterozygote advantage. Individuals who are, so let's just use S for the normal gene. Actually, let's use H for the normal hemoglobin gene. So individuals who are big H, they have normal hemoglobin. They don't get sickle cell anemia. But what they might end up getting is they might contract malaria and die of malaria. Individuals who are little h, they end up with sickle cell anemia, which is deadly itself, at least before we develop treatments for it. But individuals who are big H, little h, they not only don't have sickle cell disease, but they also get some resistance to malaria. So in places where there's a lot of malaria, these individuals are the most fit. They're the ones that are selected for. So that means that this gene for sickle cell disease is really common in places, in people who come from Africa, because in populations from Africa, because that gene in that environment was selected for because of the protection and the resistance that it conferred to malaria. So heterozygotes have an advantage over either type of homozygote. Epistasis is another non-Mendelian inheritance pattern. This is a condition in which a gene at one location on a chromosome affects a gene at a second locus or second location on a chromosome. Albinism in humans is a really good example of this. Albinism happens, it arises when individuals inherit two defective copies of the allele that normally codes for an enzyme necessary for melanin synthesis. Even though alleles at other locations, which essentially code for the amount of melanin that you end up with in your eyes or your skin or your hair, may code for having brown eyes or brown skin or brown hair. These individuals are ultimately albinos because they lack the ability to produce the pigment, in this case, melanin. So a gene at one location affects genes at other locations. That's epistasis. Final pattern we'll look at is polygenic inheritance. Polygenic inheritance occurs when a trait is actually controlled by two or more sets of alleles. Mendel thought that one set of alleles controls each trait. We know today that's not true. Examples of human traits that are polygenic include height, skin color, and the prevalence of diabetes. Each individual possesses a copy of all of the allelic pairs, each pair of alleles. These may even be located on different chromosomes. Each dominant allele has a quantitative effect on the phenotype, and the effects from all of those different pairs are additive. Populations typically exhibit continuous phenotypic variation for polygenic traits. What we mean by that, if the frequency of the different phenotypes were graphed in like a bar graph, the graph would look like a bell curve. Human skin color is thought to be controlled by three pairs of alleles. That we'll call them the A, the B, and the C alleles. A person with all dominant alleles in each pair is really, really dark-skinned, whereas a person with all recessives is very, very light-skinned. And a person in the middle with some dominants and some recessives can have an intermediate skin color. So this graph and Punnett square show us sort of how the situation works. And we can see that the phenotypes exhibit sort of this bell curve frequency. There are a few individuals that are really, really light complected. There are a few that are really, really dark complected, but most individuals end up somewhere in the middle. So again, anytime we get this bell curve or normal distribution of phenotypes, that probably is going to indicate to you that a trait is polygenic. It's controlled by more than one set of alleles. All right, one other thing I forgot about are multifactorial or complex traits. These are traits that are controlled by multiple genes, and they're also affected by environmental influences. Traits in humans like hypertension, high blood pressure, diabetes, schizophrenia, and also certain kinds of allergic conditions are probably complex or multifactorial traits. And then one final one are lethal alleles. There are certain allele combinations which are lethal and prevent the birth of individuals with certain genotypes. A good example is achondroplasia. It's the most common type of dwarfism in humans. Turns out it's actually autosomal dominant. Individuals with the genotype big A, little a are dwarfs. Individuals with the genotype little a are normal height. Individuals though, the genotype big A is lethal. Individuals with this genotype die before birth. So let's say we had two dwarfs and they were both big A, little a in terms of genotype. We draw a Punnett square for them. So big A, little a, big A, little a. The Punnett square would predict that all three of these individuals would be dwarfs and that this one would be a person of normal height. So the predicted phenotypic ratio is 3 dwarfs to 1 normal height. But remember, the big A allele combination is lethal. Those children are never born. So the actual birth ratio is 2 dwarfs to every 1 normal height person. So due to the lethality of that big A genotype, the actual probability of a living dwarf child from this cross is 2 out of 3. All right. So those are all examples of exceptions to Mendel's laws, or we might call them non-Mendelian inheritance patterns. Hope that helps.